I'm Bob Lefkowitz, an investigator of the Howard Hughes Medical Institute and a professor of medicine and biochemistry at Duke University Medical Center in Durham, North Carolina. Today I'm talking to you about drug and hormone receptors, and in particular about one remarkable, huge family of such receptors called the 7 transmembrane receptors, also known as the G protein coupled receptors. In my first lecture, I told you in some detail about the history of how this field evolved and how we traveled from being quite skeptical about the mere existence of these receptors to a fairly detailed understanding of how they function. Today, I want to bring you right up to the minute, talking about interesting and exciting discoveries over the last 5 to 10 years, which are completely reshaping our understanding of how the receptors function and are regulated, and which can be leveraged to develop entirely new types of drugs. The focus is going to be on a class of molecules called beta-arrestins. Now, I introduced this in the first lecture, but to just briefly review, the classical signaling paradigm for understanding how G protein coupled receptors work is as shown here, and was derived over the years largely from studies on 2 model systems: the beta-2 adrenergic receptor for catecholamines like adrenaline and noradrenaline, and the so-called visual receptor, rhodopsin, for which the stimulant is really photons of light. Now, the key feature as shown here is that when the receptor is activated by adrenaline or by a photon of light, it changes conformation, becomes activated, stimulates G proteins, leading to activation of second messenger generating enzymes, and those second messengers activate kinases, leading to cellular responses. But as we talked about, an opposing system immediately is engaged, which tends to shut down or desensitize receptor action. This involves a 2-step mechanism. In the first step, a G protein coupled receptor kinase, such as GRK2 shown here, phosphorylates the active conformation of the receptor, leading to a second step in which a beta-arrestin molecule binds to the phosphorylated receptor, physically blocks G protein coupling, sterically interdicts it, thereby leading to desensitization. But the exciting thing in this field, which has developed over the last few years, is that an entirely new signaling paradigm is emerging, and that is shown here. The notion is that the GRK beta-arrestin system is really bifunctional. At the very same time that it desensitizes G protein signaling, it also serves as a signaling unit, a signal transducer in its own right, which is able to lead to signaling to a growing list of biochemical pathways, some of which are listed here, which in turn lead to a variety of important cellular physiological consequences. This system also can lead to endocytosis of the receptors through clathrin. So today we'll be talking about this new appreciation of the signaling and other functions of beta-arrestins and how this can be leveraged for drug discovery. Let's start with the first, or core function, of the beta-arrestins, which I've told you about already. Their ability to sterically block G protein signaling, thereby slowing the rate of second messenger generation. But quite recently we discovered that this mechanism is actually more complex. At the very same time that the arrestins are slowing the rate of G protein activation, they also serve as adapters which can recruit the very enzymes responsible for the degradation of the second messenger. Now, in the case of the beta receptor in GS, which leads to cyclic AMP stimulation, that would be the cyclic AMP phosphodiesterase, shown here. In the case of a GQ-coupled receptor, like the muscarinic-cholinergic receptor, which leads to diacylglycerol formation, that would lead to the recruitment of diacylglycerol kinase by beta-arrestin. Now, this function essentially serves to bring these degradative enzymes into close proximity with those microdomains of the plasma membrane where the second messenger is being generated. So this new information suggests that even the core function of the beta-arrestins, the desensitization, is actually more complex than we originally perceived. Specifically, it involves a concerted action whereby the beta-arrestins both slow the rate of second messenger generation while enhancing the rate of degradation. Now, the second function of the beta-arrestins is to serve as adapters which facilitate the clathrin-mediated endocytosis of the receptors in response to agonist. This core function requires the ubiquitination of beta-arrestins by E3 ubiquitin ligases, MDM2 in particular is important here, and that leads to the interaction of B-arrestins as an adapter, not only with clathrin, but with the clathrin adapter AP2, and with a growing list of other elements of the endocytic machine. Now, several years ago, we were carrying out a proteomic screen, which I'll tell you more about in a few minutes, looking for molecules which bound to beta-arrestins. And in the course of doing that screen, Kevin Zhao in our group discovered that a variety of cellular motors bound to the beta-arrestins, in particular, as one example, the kinesin Kif3A. This led to a very interesting line of research in our laboratory by Jeff Kovacs, which involves smoothened. Now, smoothened, as some of you may know, is a very atypical, non-canonical, if you will, 7-transmembrane receptor, which may not even couple to G proteins at all. Together with its inhibitory co-receptor patched, it mediates the important effects on the ligand of the ligand sonic hedgehog to lead to cellular proliferation and embryonic pattern formation. Now, several years ago, a number of laboratories showed that in order for smoothen to function, it must be translocated to the primary cilium of cells so that it can be in close proximity with the Angll family of transcription factors. Now, since it was known that the kinesin Kif3A was necessary for primary cilia formation, and moreover, that it was somehow, by unknown mechanisms, required for smoothen function, and given that we had discovered that there was an interaction between B-arrestins and Kif3A, we wondered whether this interaction of B-arrestin and Kif3A might somehow be involved in the translocation of smoothened, which was required to the cilia in order for it to function. And in fact, we were able to find that when the system is stimulated by sonic hedgehog, B-arrestin 2 is recruited to smoothened, thereby bridging a ternary complex of smoothened, B-arrestin, and Kif3A. The Kif3A motor then translocates the entire unit to the primary cilium into proximity with Angll transcription factors, which can then be activated, leading to smoothened signaling. And in fact, we demonstrated, as has Mark Caron's group, that in this case, B-arrestin 2 is required for smoothened signaling. Interestingly, GRK2 appears to be required as well. These interesting and somewhat unexpected results have raised the possibility that B-arrestins might function even more widely as adapters to motors to facilitate the cellular trafficking of other G protein coupled receptors and perhaps for other cellular cargoes as well. Now, the most recently appreciated function of the beta-arrestins, and one which I'll be spending the most time on, is their ability to serve as signal transduction units in their own right. And this was first discovered about a decade ago for the non-receptor tyrosine kinase C SRC, but subsequently has been very widely studied for MAPK kinases of various types, AKT, and a growing list of other signaling systems. Again, ubiquitination of B-arrestins appears to be necessary for these functions. My laboratory in recent years has been particularly interested in learning about other pathways which are activated through this mechanism, and also what pathways or consequences might lie downstream of activation of the pathways we already know about, specifically the MAPK kinases. Even more remarkably, it turns out that the B-arrestins can serve as signaling adapters, not just for G-protein coupled receptors, but it is turning out for specific examples of receptor tyrosine kinases, like the IGF receptor, cytokine receptors, ion channel receptors, such as the nicotinicholinergic receptor, and even Notch. Now, in the course of studying the signal transduction properties of beta-arrestins, we were led to an interesting discovery which has turned out to have markedly facilitated this aspect of the work, while at the same time serving to revise in significant ways some of our most fundamental understanding of how receptor biology works. And as you'll see toward the end of the lecture, has actually led to a new approach to developing therapeutics. I'm speaking here of our discovery of what we refer to as biased agonists. Now, a biased agonist is a ligand which stabilizes a particular active conformation of a receptor, thus stimulating some responses but not others. 7 transmembrane receptors, for example, can be biased toward a particular G protein or beta-arrestin. Mutated receptors can also be biased. Now, I told you before in the first lecture that the classical core concept, central dogma, if you will, of pharmacology is that receptors can exist in inactive R and an active state R star, and that agonists, here displayed as A, stabilize or induce the activated conformation, with all signaling consequences then being a downstream result or a function of the concentration of active R star. But the existence of biased agonists, which I've just described to you, immediately says that this is too simple, that there must be multiple active states of the receptor, at minimum, for this purposes of this discussion, 2, one of which mediates G protein signaling, and the other which would mediate B-arrestin signaling. And presumably there are a significantly larger number of active states than 2, but of course we can't know how many at this point. Let me illustrate this idea with some data about the very first biased agonist and biased receptor which we discovered. And these both relate to the actions of angiotensin, the most potent vasoconstrictor which is made in the human body. We had read about 2 reagents which seemed interesting to us based on our own previous work. One was a mutated angiotensin octopeptide, which is referred to as ß-arrestin for the 3 mutated residues in the polypeptide chain. The second one was a mutated receptor, angiotensin receptor, shown here, which had mutations in 2 of the 3 highly conserved residues, which I mentioned to you in the first lecture, the so-called dry, DRY motif. 2 of these are mutated to alanines, and this is called the dry AAY receptor. Now, the common feature of the mutant angiotensin peptide and the mutant angiotensin receptor is that they are unable to activate G protein signaling, but both can lead to internalization. These interested us because, as I've told you, we were aware of the fact that B-arrestins are able to serve as adapters which mediate endocytosis of the receptors. So we obtained some of the peptide, had it synthesized, and we obtained the receptor mutant, and we looked at their ability to activate G protein signaling. Now, what you can see here is that in confirmation of results which had been previously published in the literature, neither the mutant receptor nor the mutant peptide, when applied to a wild-type receptor, could lead to G protein signaling as assessed in a classical second messenger generation. So here we're looking at PI turnover, and when a wild-type angiotensin receptor is stimulated with wild-type angiotensin, a classical dose-response curve is observed, but absolutely no activity for either the mutant peptide applied to a wild-type receptor or the mutant receptor stimulated with wild-type angiotensin. We also found no calcium release, no GTP gamma S binding to G proteins. So clearly, as reported, the mutant peptide and mutant receptor are devoid of G protein stimulating activity. However, both are very robust in their ability to recruit beta-arrestins. Now, shown here are confocal micrographs. In this upper panel are shown HEK cells which express the angiotensin receptor and which are also expressing GFP beta-arrestin 2. As you can see, the B-arrestin is diffusely expressed in the cytoplasm but is excluded from the nucleus. When such a cell is stimulated with wild-type angiotensin, 20 minutes later, the B-arrestin 2 GFP can be found concentrated in these endocytic vesicles. That beta-arrestin had first been translocated to the plasma membrane, which we would have seen at an earlier time point, and has now been internalized. What you can't see is that the receptors are also co-localized with it. However, both the mutant peptide, when applied to a wild-type receptor, or the mutant receptor stimulated with angiotensin, also robustly recruit B-arrestin and lead to its co-localization with receptors in these endocytic vesicles. This despite their complete inability to activate G protein signaling. So clearly, these mutant peptide and receptor are devoid of G protein stimulating activity, but do have robust ability to recruit beta-arrestin. Now, of course, the question becomes, can they use that B-arrestin as a means of signaling into the cell? And to initially look at that, we chose a highly conserved signaling system, which I'm sure is familiar to most of you, namely the MAP kinase cascades. As you recall, the MAP kinase cascades are highly conserved from yeast to humans. They consist of 3 kinases in sequence: a MAP kinase, a MAP kinase, and a MAP triple kinase. The MAP kinases in turns are in several families, such as the ERKs, the JUNKs, and the P38 MAP kinases. There are at least a dozen or so kinases at every level of this cascade. So there's enormous complexity. Many different types of G protein coupled receptors activate these pathways, as do all sorts of other growth factor receptors, cytokines, etc. And of course, the classical pathways that have been worked out involve phosphorylation by the MAP kinases of transcription factors leading to their activation, translocation to the nucleus, and regulation of all manner of transcriptional programs shown here. Now, a conundrum during the 90s which was investigated, was the question of how, with all this complexity, with all these welter of enzymes, dozens of enzymes, how did the cell ever organize any specific pathway, such as, for example, RAF, MEK, ERK1? How did it ever organize such a pathway with any fidelity or reproducibility? And this led to the idea that there might be scaffolding proteins, which would bind together several members of a particular pathway. And in yeast, a molecule, sterol 5, was discovered, which appeared to function in this way. We, in fact, were able to demonstrate that beta-arrestins, particular beta-arrestin 2, was able to serve as a scaffold for several of these MAP kinase pathways. For example, it was able to scaffold a RAF, MEK, ERK pathway, and an ASC, MKK4, JNK3 pathway. So it was able to serve as a scaffold for such pathways. Let me show you one of the key experiments which led to these types of discoveries. This shows that in terms of ERK MAP kinases, angiotensin gives robust activation of ERK MAP kinases in a cell which has been transfected to express the angiotensin receptor. These are HEK cells. However, the ß-angiotensin, when applied to this cell, also leads to ERK MAP kinase activation to the tune of about 50% of the wild type. And the same is true for the mutated angiotensin receptor. Now remember, in this case, there is absolutely no G protein activation, and nonetheless, we're getting half as much ERK activation assessed by a simple assay of phospho or activated ERK. We're getting half as much ERK activity as we did with the wild type system. Now, if we use RNAi to knock down beta-arrestin 2, we lose about half the activity of the angiotensin-stimulated system. But we completely ablate the activity of siI-angiotensin or the mutated receptor, suggesting that they are activating the ERK system exclusively via beta-arrestins. In the case of the wild-type system, if we block the G protein arm with a PKC inhibitor, this activity goes to zero. So again, even though there's no G protein activity here, we are getting B-arrestin-mediated activity. So to summarize, many more experiments by my laboratory and other laboratories working now with dozens of different types of G protein coupled receptors have led to this type of understanding. In the case of a GQ coupled receptor, such as the angiotensin receptor, it appears to be able to activate this specific effector system, ERK-MAP kinase, by two distinct, parallel, and independent pathways. One is the classical G protein pathway, leading to second messenger generation, ERK activation, and translocation to the nucleus to regulate transcriptional programs. In the case of the B-arrestin pathway, in this case B-arrestin 2, this leads to activation of a distinct pool of ERK, which is sequestered and restrained exclusively in the cytosol and prevented from going to the nucleus. This undoubtedly leads to the activation of distinct cytosolic substrates with distinct cellular physiological consequences. We've been able to demonstrate this biology, as have other laboratories now, very analogous biology with quite a large number of other receptors. As I said, the mechanism appears to be a physical scaffolding of the different components of the ERK-MAP kinase. Now, in our effort to understand the downstream consequences of this B-arrestin-mediated signaling, in this case through ERK, but other pathways as well, we've recently been using a variety of techniques. Some of them are shown here. We use these B-arrestin-biased ligands, which I've already mentioned to you, RNA interference, knockout cells from B-arrestin 1 or B-arrestin 2 knockout animals, the knockout mice themselves, and as I will show you, a variety of proteomics techniques. What I'd like to do is give you several examples, just examples of novel pathways, B-arrestin-mediated pathways, which we've discovered just in the past several years. So for example, we have found a mechanism which operates downstream of the beta-1 adrenergic receptor in cardiomyocytes or the angiotensin receptor in vascular smooth muscle cells. It leads to transactivation of the EGF receptor and consequent activation of ERK, and it goes through a B-arrestin-mediated activation of SARC, then of metalloproteinases, which release EGF receptor ligands like HBEGF. This pathway appears to be cardioprotective. We found in vascular smooth muscle cells that through a B-arrestin-mediated activation of ERK-MAP kinases, angiotensin is able to lead to ERK-mediated phosphorylation and activation of a cytosolic kinase called MINC, which phosphorylates and activates EIF4E, a rate-limiting step in protein translation, leading to an increased rate of protein synthesis. We found that in vascular smooth muscle cells, again, the angiotensin receptor, operating through B-arrestin-mediated signaling, is strongly anti-apoptotic. And this anti-apoptotic activity is mediated through a concerted mechanism in which the B-arrestins activate ERK and PI3 kinase AKT, two survival pathways, both of which lead to and converge on phosphorylation and an activation of pro-apoptotic BAD. Note that the ERK arm proceeds through phosphorylation of P90 risk. Finally, an interesting pathway, which I'll say more about in a clinical context momentarily, operates downstream of what was previously an orphan receptor, GPR109A, now known to be a receptor for the very important cholesterol-lowering drug niacin. This pathway leads through beta-arrestin 1 to activation of PI3 kinase and AKT, and then on to activation of phospholipase A2, generation of arachidonate, and vasodilatory prostaglandins. And I'll have more to say in just a few moments about the therapeutic relevance of this particular pathway. So this little cell I've constructed here, with each of the pathways I've just mentioned put into that cell, should begin to give you an idea of the rapidly growing complexity of these pathways and networks, signaling networks, which operate downstream of B-arrestin. Here are just a few pathways delineated by my laboratory in the past several years. If we were to graft onto this all of the currently known B-arrestin-mediated signaling pathways, it would be too complex to even look at. And what this then has suggested is that we need some other way of looking at this issue of B-arrestin-mediated signaling. I mean, if we have any hopes of trying to understand the much broader universe of cellular signaling that's mediated through B-arrestin, we really need to take a more general and global approach. And so that is the direction in which we have begun to move, because I think this type of work has clearly now suggested that signaling downstream of B-arrestins is undoubtedly probably every bit as complex and multifaceted as that downstream of G proteins. And so in this regard, we've begun using a variety of global proteomics approaches and systems biology approaches. So in our first effort, Kevin Zhao and our group conducted a proteomics screen to try to find out or delineate the universe of proteins bound to beta-arrestins, either constitutively or after stimulation of a G protein coupled receptor. And again, the model we used in our initial experiments was the angiotensin receptor. So HEK cells expressing the angiotensin receptor were stimulated or not with angiotensin, and then B-arrestins were immunoprecipitated, and mass spectrometry analysis was used to define the proteins that were bound. In addition to obtaining really most of the dozen or so known interactors that we had previously discovered on a case-by-case basis, Kevin was able to discover dozens and dozens of novel interactors. Perhaps not surprisingly, many of these fell into the category of signal transduction proteins. And then, of course, we had motor proteins and several other groups. Amongst the signal transduction proteins were a variety of protein kinases and phosphatases, trafficking proteins, small GTPases, etc. Many fascinating interactors which serve as springboards for discovery. And then, as I mentioned, we found several families of motor proteins, including, of course, Kif3A, which I've already talked to you about. Even more recently, we've begun doing a quantitative global phosphorylation analysis of B-arrestin-mediated signaling. The procedures here are as follows. We take a cell expressing a GPCR, such as, for example, the angiotensin receptor in vascular smooth muscle cells. We then stimulate the cell with a B-arrestin-biased ligand, such as siI-angiotensin. Now remember, this will activate only B-arrestin signaling, not G protein signaling. We can document that by then repeating the experiment after siRNA knockdown to eliminate B-arrestin-mediated signaling. We then use SILAC technology, stable isotope labeling of amino acids in cells, to quantify all the phosphorylation sites in the cell before and after stimulation. And then the phosphopeptides are characterized by tandem mass spectrometry. Now, in some of our earliest results, again using SII-biased, B-arrestin-biased ligand stimulation of vascular smooth muscle cells, Kevin was able to identify 25,000 individual phosphopeptides derived from about 2,500 total phosphoproteins. Now, Not surprisingly, and in fact gratifyingly, most of those don't change when you stimulate with siangiotensin. You wouldn't expect them to. In fact, about 85% of those phosphorylation sites do not change. However, about 15% do. 10% go up, and about 5% go down after stimulation of B-arrestin-mediated signaling. Some of the changes are very dramatic indeed. But of course, the problem is, now that one has these huge piles of data, literally, these long lists of proteins and how much the phosphorylation is changing, the question is, how does one begin to organize and analyze these vast quantities of data to understand what they're trying to tell us about B-arrestin-mediated signaling? And so in this regard, we've been increasingly turning to the techniques of systems biology, using a variety of commercial pathways, commercial packages for pathway analysis, as well as working with several collaborators, Avi Mayan at Mount Sinai and others at Duke, trying to understand and explore the best ways of analyzing this data. I wanted to give you one specific example, just to show you how this is working, of a particular network which we have pulled out as being very enriched in the B-arrestin signaling universe. And this is a network for cytoskeletal rearrangement. Now, cytoskeletal rearrangements are extraordinarily complex in terms of the biology and involve the concerted interaction, literally, of dozens of proteins. Now, in this slide, and this is unpublished data, rather than listing the names of individual proteins, we've just given them numbers. But suffice it to say, this is a reproduction with the names of the proteins removed of currently understood signaling network which is involved in cytoskeletal rearrangements which occur, for example, in cell motility during chemotaxis. Now, every protein colored red here was identified as being enriched in either the B-arrestin interactome or in the signaling phosphoproteome I just told you about, or both. So clearly, many of these proteins involved in this cytoskeletal rearrangement network are being engaged, phosphorylated or dephosphorylated in response to B-arrestin signaling. Further, those which are circled in green here have already been validated in our laboratory on a case-by-case individual basis by techniques such as co-immunoprecipitation or others. So these are very early days now for us in terms of these types of approaches. But hopefully what I've just shown you here can give you a sense of how we're proceeding with this aspect of the endeavor. Over the next few years, we plan to apply this type of analysis to several different receptors in several different cell types. I also want to point out that exactly the same methodology and approaches can be used to explore B-arrestin-mediated signaling, which plays out not just through phosphorylation, but through other types of post-translational modifications, such as, for example, nitrosylation or ubiquitination. Okay, so we've reviewed then the interactions and the functioning of the B-arrestin and GRK system to mediate desensitization of the receptors by several different concerted mechanisms, their ability to serve as adapters for clathrin-mediated endocytosis, and now this burgeoning area of signal transduction and scaffolding of signaling proteins. But one of the most important implications of this rapidly growing area is that there are some important therapeutic implications, and I want to begin to introduce that concept now. And that's shown in this cartoon. The idea, as I've shown you, is that a 7-transmembrane receptor can signal in two distinct ways, either through G proteins or through B-arrestins. However, in certain pathophysiological circumstances, G protein-mediated signaling can have deleterious consequences. For example, stimulation of the angiotensin receptor physiologically through G proteins can raise blood pressure. Not a good thing in a patient with heart disease. Similarly, beta-adrenergic catecholamines, adrenaline, noradrenaline, working through the beta receptor, can stimulate the force and rate of beating of the heart. Not a good thing for a patient with effort-induced angina. On the other hand, as I've showed you, some of the recently discovered signaling through beta-arrestins may have beneficial effects, such as, for example, an anti-apoptotic action. Now, let's talk about angiotensin receptor blockers, or ARBs. Angiotensin receptor blockers are some of the most useful drugs, and certainly the most widespread used drugs in cardiovascular medicine today. Their utility specifically relates, as does that of beta blockers, to their ability to block potentially deleterious G protein mediated effects, in the case of ARBs, angiotensin, for example, through to raise blood pressure. On the other hand, ARBs block not only these deleterious G protein effects, but they also block all effects mediated through B-arrestin, effects which, one, weren't even known till the last few years, and two, which, as I told you, may be beneficial. So we hypothesized that perhaps a drug like ß-angiotensin, which will block G protein signaling, which it does quite effectively because it occupies the binding site and doesn't signal through G proteins, but which, unlike conventional ARBs, is able to also stimulate B-arrestin-mediated signaling rather than blocking it, which is what would happen with a conventional blocker. We speculated that this might represent a new type of drug. And in fact, it turns out that ß-angiotensin is able to slow the progression of heart failure in various animal models of heart failure. It lowers blood pressure and is in fact anti-apoptotic. In a very analogous fashion, we have screened almost 20 beta blockers, representing almost all beta-adrenergic blockers used in the world today to treat heart disease. And the question we asked was, were any of these blockers able to actually signal through B-arrestins while blocking G protein activation? Only a single one showed this capability, and that is carvedilol, or Coreg, which, in addition to its ability to potently block G protein signaling, was able to weakly stimulate through B-arrestins. In fact, carvedilol has unique efficacy in treating congestive heart failure, is anti-apoptotic and cardioprotective. Another interesting example of a B-arrestin biased ligand is shown here for the parathyroid receptor. This particular PTH analog has been known for years as a powerful competitive antagonist of parathyroid hormone. In fact, it's an inverse agonist. It actually lowers the activity basally of the parathyroid hormone receptor. Now, you'll recall that parathyroid hormone is a key regulator of calcium metabolism and a key stimulus for anabolic bone formation. In fact, it is used clinically to treat osteoporosis. Well, several years ago, Diane Guesty-Palmer in our laboratory discovered that this analog, while it is in fact an antagonist of G protein activation through the parathyroid hormone receptor, is actually a B-arrestin biased ligand which is able to activate, for example, in cells like osteocytes, is able to activate ERK-MAP kinase activity through the PTH receptor. Recently, quite remarkably, she found that when this analog was administered to mice, it led to anabolic bone formation, even though it can't activate G proteins at all. And remember, the textbook explanation for the activity of parathyroid hormone to build bone is that this is exclusively a GS and cyclic AMP mediated process. Well, she found that administration of this ligand, which actually blocks G protein activation, but which stimulates B-arrestin-mediated signaling, leads to anabolic bone formation without bone resorption. Moreover, this effect was completely lost in B-arrestin-2 knockout mice. So, this B-arrestin biased ligand for the PTH receptor may represent a new therapy for osteoporosis. Now, biased ligands don't need to be biased toward B-arrestin to have potential therapeutic utility. There are currently several very provocative examples where a G protein biased ligand might well serve novel therapeutic purposes. Let me give you a couple of examples. I mentioned before that niacin, vitamin B3, administered in therapeutic doses, like several grams a day, is one of the most effective, probably the most effective agent we currently have available to us for raising good HDL cholesterol and lowering triglycerides. It also more weakly lowers LDL, but is not as effective in that regard as statins. The problem with this potentially wonderful cholesterol-modifying drug, niacin, is that very few patients can tolerate it because it leads to severe, painful flushing, which is a result of the release of vasodilatory prostaglandins. I also mentioned to you in the first lecture and earlier that the biological effects of niacin are mediated through this previously orphan receptor, 7-TM receptor, GPR109A. In fact, in knockout mice in which this receptor is deleted, both the beneficial therapeutic effects on lipids as well as the very annoying flushing are both lost. Well, in our lab, Rob Walters recently was able to show that the lipid-modifying effects of niacin are actually mediated through G protein activation, whereas the flushing, which is basically quantitated in mice by using a laser Doppler apparatus to monitor vasodilation in the earlobe, he demonstrated that that effect is mediated through beta-arrestin-1 signaling and not through the G proteins. In particular, it utilizes the pathway I showed you before, which leads from GPR109 through arrestin-1 mediated activation of phospholipase A2, arachidonate, and vasodilatory prostaglandin release. In fact, the vasodilatory response to niacin was largely ablated in beta-arrestin-1 knockout mice, although the lipid-modifying effects, the desired therapeutic effect, is retained. This suggests then that a G protein biased ligand, niacin analog, for GPR109A might represent a novel and very effective form of therapy for cholesterol problems, which would not lead to the very distressing flushing and which could then be tolerated by many more patients. Now, another example of a potentially useful G protein biased ligand would be one for the opiate mu-opioid receptor. Now, of course, we all know that opioids are the single most powerful anti-nociceptive, analgesic, pain-relieving medicines that we have. However, they cause a number of distressing side effects which limit their utility. Most obvious is tolerance, which requires more and more drug to get the same effect, but also side effects such as constipation, respiratory depression. Now, the desired anti-nociceptive effects of opioids are mediated through stimulation of the GI proteins through the mu-opioid receptor. But recently, Laura Bahn showed that tolerance to opiates is markedly diminished in B-arrestin-2 knockout animals. B-arrestin-2 interaction with the receptor causing desensitization appears to be the key mechanism of the tolerance. So that's lost in B-arrestin-2 knockout animals. And the constipating and respiratory depressing effects of opiates are also markedly depressed in B-arrestin-2 knockout animals. So this then suggests that the desired therapeutic effects of the opiates go through the G proteins arm, whereas the tolerance and other side effects go through the B-arrestin-2 arm. And this then leads to the supposition that a G protein biased ligand for the mu-opioid receptor would be a particularly useful agent in treating pain, and one which would represent a significant advance over therapies that are currently in hand. So to summarize then, these several examples of B-arrestin biased ligands and G protein biased ligands should give you a sense of how this newly acquired knowledge of these distinct signaling pathways can be leveraged for therapeutic gain. Now, the last topic I want to touch on has to do with trying to understand some of the biophysical basis for these distinct G protein and B-arrestin biased signaling mechanisms. Now, from what I've told you before, it should be clear then that receptors such as the angiotensin receptor or other 7-membrane spanning receptors can exist in at least 2 conformations, probably more, shown by these differently shaped squiggles on the C-tail of the receptors. Now, one of these conformations presumably leads to G protein activation, whereas the other leads to, for example, B-arrestin-mediated signaling. An unbiased ligand, such as angiotensin, appears to be able to do both, whereas a biased ligand, such as SII, can only do one, either the B-arrestin or the G protein arm. So we wondered whether B-arrestin biased ligands, whether B-arrestins themselves might be able to also assume distinct conformations, conformations which would correspond to the distinct conformations of the receptor and which would then lead to distinct functional outcomes. And so to test this idea, we've recently been using a very interesting probe which is shown in this animated slide right here. Now, as you can see, this is an intramolecular beta-arrestin 2 BRET biosensor. This has been genetically modified. It was originally developed in Michel Bouvier's laboratory in Montreal. At the amino terminus is renylluciferase, and at the C terminus is YFP. Now, when substrate cilintrazine is added, bioluminescence is generated by the luciferase, which, if in the correct orientation and close enough, usually said to be less than 100 angstrom, is picked up, the energy picked up by YFP and emitted as yellow light. Conformational changes induced in the B-arrestin biosensor will modify the distance and orientation such that the BRET ratio will change. In our initial experiments, we stimulated cells through the angiotensin receptor and looked at how that was reported by the B-arrestin BRET biosensor. So in the initial set of experiments, we had the angiotensin receptor stimulated by the unbiased ligand angiotensin. When we did this, we observed an increase in the BRET signal, presumably representing the 2 ends of the molecule being closer together or in a more favorable orientation. In contrast, when we stimulated with the B-arrestin biased ligand SII, we observed a decrease in the BRET ratio. Both of these effects could be blocked by the competitive angiotensin receptor blocker valsartan. Then we used the B-arrestin biased receptor, that angiotensin receptor dry AAY, which I told you about before. Now you can see that with the B-arrestin biased receptor, both angiotensin and SII led to a decrease. So these, and again, both were blocked by valsartan. So these directionally opposite changes in the BRET ratio by the biased and unbiased ligand clearly indicate that they must be inducing distinct conformations in B-arrestin, conformations which may well be associated with their distinct biological profiles. We have observed similar results now with probably half a dozen different receptors, using both biased ligands and biased receptors. Here's an example for the PTH receptor. I told you about that B-arrestin biased ligand before. And with the parathyroid hormone receptor, stimulation by the unbiased PTH leads to an increase in BRET ratio, where stimulation with the biased PTH ligand leads to a decrease. So again, the variations are similar to what we observed with the angiotensin receptor. So this animated cartoon, with which I will conclude, demonstrates these distinct conformations of beta-arrestins which correspond to the distinct conformations of the receptor, shown by the differing shapes of the receptor. A biased ligand, such as SII, leads to a conformation of the beta-arrestin, in which the two ends of the molecule appear to be moving apart, thereby leading to the decrease in BRET ratio. Whereas shown in the upper panel, when an unbiased ligand is used, we now get an increase in BRET ratio corresponding to a moving together of the two ends of the molecule. So clearly the biased and unbiased ligands are leading to distinct conformations of the receptor, and this would suggest the beta-arrestin. Now going forward, the work in many laboratories around the world is now focused on trying to use methods such as X-ray crystallography and other biophysical techniques to document the detailed interactions and conformational changes which are characteristic of the distinct functions of B-arrestins and the receptors. And as I mentioned to you before, this is a big interest in the case of receptor structural biologists as well, who are now interested in learning about the details, the conformational details of those receptor conformations which lead to G proteins as B-arrestin or B-arrestin-mediated signaling. And hopefully in a few years I'll be able to stand before you again to present the results of such experiments.